Hello and welcome back to Chapter 2 in the video series The Birth of Genetics and the Study of Heredity. In this chapter, we are exploring the problem of heredity before genetics even existed. In the previous episode, we dived into 18th century England and watched Robert Bakewell reshape livestock through selective breeding, long before genes were understood. Now we move forward in time and we cross the channel. We are in the middle 19th century French fields. As heredity was mysterious in Bakewell's sheep and cattle, it was also elusive in agricultural plants. By the 1800s, plant breeders were facing the same fundamental puzzle. Traits could persist or vanish across generations, but no one could explain how. And in France, one seed company became an unexpected bridge between agriculture and experimental science, the Vilmorin Company. Between 1840 and 1920, nearly a century after Robert Bakewell's experiments, the Vilmorin Seed Company had become one of the most important producers of cereal seeds in France, especially in the Paris region. But the Vilmorin Company was more than a simple commercial enterprise. In fact, it actively promoted experimental research in plant breeding. For the Vilmorin, plant improvement was not simply about selling better performing plants. Their work became a systematic investigation into variation, transmission, and the persistence of traits across generations. In other words, by the mid-19th century, heredity was no longer something that farmers just used. It had become an experimental problem. The foundations of the company were laid by Philippe de Vilmorin, who was born in 1776 and died in 1862. And he was Louis's father. In the 1840s, Philippe made quite a bold claim, namely that domesticated varieties would arise from wild plants just in few generations. He argued that within just 3 generations, a wild carrot could be transformed into something looking like a domesticated or also said cultivated root. This view challenged the dominant idea that domestication required a slow, gradual change. The broader story of plant and animal domestication is far more complex, and we will explore it properly in another episode. But what matters for now is Philippe's claim: change in plants could be accelerated by deliberate selection. So Philippe's insight was not speed, it was deliberate selection. He insisted that improvement could not just come from a better soil or better care, also named the soin in French. Improvement, according to Philippe, depended on the judicious choice of the seed bearer. In other words, improvement had to act through the seed. Now, whether Philippe's experiments involved mass selection, which means he selected many desirable plants and bulked their seeds together, or he performed lineage selection, keep the seeds of each individual plant lineage separate and track their descendants, remains unclear. But what mattered was this: he understood that improvement depended on transmission, and that material transmission differed between individuals. It was Louis de Vilmorin, Philippe's son, who turned his father's ideas into a systematic method in the 1850s. He developed further on the theme and expressed what he called the selection process of the seed bearer, later known as genealogical selection or also pedigree selection. This one was a major innovation in plant breeding. Instead of selecting the best plants from a population and mixing their seeds together, Louis isolated seeds from single plants and kept their descendants separate. In this way, each lineage was grown independently, and each family was evaluated generation after generation. The goal here was not simply of finding good individuals. The goal was to identify plants that were capable of transmitting desirable traits with constancy. In modern terms, this was an early attempt to distinguish the plant's genetics from the condition it grew in, the so-called environment effect. Louis de Vilmorin even described this as establishing a kind of nobility in plants. It may sound funny, but what he said was that a plant had to prove itself a good stallion, un bon étalon, which meant a plant, an individual that was capable of passing its qualities reliably. And its rule was to never pull the seeds. The genealogy had to be tracked from the beginning of the experiment. So we can say that at this point, heredity was not simply assumed. At this point, it was monitored. One example of Louis de Vilmorin experiments is the experiment that he carried out in the 1850s on sugar beet. What he did was to select between 20 and 30 offsprings from each plant lineage. He focused at this point on quantifying sugar content, which by those times was measured by extracting the juice from the beet and assessed it chemically. In this way, he provided numbers of what was later called phenotype, the external characteristics of the trait, and these numbers were used to compare the progenies. Louis measured two things: the average sugar content of each lineage and the variability of the sugar content in each lineage, which in this case would be the sum of the variation of the unit from the mean of the lineage. For Louis de Vilmorin, improvement was not just increasing the trait, but it also meant reducing the variability within the lineage. A successful line for him was one that showed fixity, namely stability across generations. This quantitative approach in genetics preceded by nearly 2 decades the statistical work of Francis Galton, which work we will hear in one of the future episodes in this series. So, long before the concept of genetics as a science existed, heredity somehow was being measured in traits. Before Mendel, heredity was often imagined as a struggle between two opposing forces. For instance, in 1847, Prosper Lucas, a psychiatrist, explained transmission of traits as the result of two opposing forces, one conserving stability, called heredity, and one producing the variation, called innateness. Louis de Vilmorin adopted a similar framework. He spoke of centrifugal forces, which generated variation, and centripetal forces ensuring stability. The centripetal force had two components itself. One was called atavism, which was this force that led an individual to resemble its earlier ancestors. And the other was the direct heredity, which worked on the resemblance to immediate parents. Atavism was a term coined in this period, and it relates to the Latin word atavus, which means ancestor. According to Louis, in the wild, atavism dominated, meaning that the variation in the wild of the species was more under control, and species characteristics remained more stable. Under domestication, so with the cultivated material, however, human selection and deliberate crosses between individuals protected some rarer forms of variation from being erased. For Vilmorin, the genealogical selection, so the following selection across a pedigree, was a way to counteract atavism. In other words, breeding was a fight against nature's tendency to return to the ancestral form. Louis de Vilmorin tested these ideas in experiments in Lupinus hirsutus, lupin, a self-fertilizing legume plant that can have blue or pink colored flowers. Because the plant self-fertilizes, each generation had a single ancestor. Henri, Louis' son, published his father's work in 1879, and he showed in his publication one of his father's schemes. Louis started his experiments with two purified sets, those seeds coming only from blue flowered plants and those seeds only coming from pink flowered plants. This purification method was across 12 generations to pick the seeds of the plants that produced blue flowers or only the seeds that produced pink flowers. Louis' experiment lasted 4 other generations after the 12th generation of purification. And during each generation, few plants were selected to provide the seeds for the next generation. What he did was to count the proportion of flower colors across different generations for each individual plant. For example, the proportion of blue flowered plants, given that the original plant came from the blue set, or vice versa came from the pink set. What he thought of doing then was of using these proportions as intensities of the forces that he earlier mentioned. What he could conclude was that the direct heredity, this force that somehow worked on the resemblance between parents and offsprings, worked on the immediate after generations. On the other hand, the atavism, the resemblance to the ancestors, increased over time, over generations. So, from a pool of blue seeds, the first plants would be most of them blue, but over generations you would start seeing pink flowers. So what he saw also was that the parental influence was weakened after the first generation. In summary, what Louis was already doing in his experiment in lupinus was counting distribution of traits across generations, long before Mendel. But he did so without a predictive mathematical law, so that heredity in his experiment was observable, but it was not yet formalized. From the 1870s onward, hybridization became central in the Vilmorin Company's programs. Crossing of, for example, wheat lines coming from different regions of France was shown to produce greater variability than spontaneous variation alone within a regional lineage. And this should recall you what we said about Bakewell's experiments in longhorn cattle. He also worked with, it is disputed, but it seems that he also worked with longhorn cattle coming from different regions of Britain in order to expand the genetic variability of his pool. But crossing between different lineages was only the beginning of their breeding programs. What happens when crossing two different lineages is that often the F1 individuals, so the offsprings, may show predominance, so may look like one parental trait mostly. To reach a certain stability in how a trait would show itself, genealogical selection was needed, or also called pedigree selection. So that initial crossing created variation, and then pedigree selection created constancy, so that the trait would keep showing itself reliably. At the same time the Vilmorin Company was working on wheat crossings, Charles Victor Naudin was working on cucurbits. The theory that he proposed was that hybrid variability resulted from the disjunction of parental essences. And this might recall you something. What he thought was that different characters separated and then recombined independently, so that he could describe hybrids as living mosaics. Naudin's insight came remarkably close to Mendel's later conclusion, but Naudin lacked what was a predictive scheme. He observed patterns but did not formulate any law. So in his ideas, the pieces were already there, but they were not yet assembled. So to conclude this second part of this chapter, why heredity was a problem before genetics, what we can say is that the Vilmorin and other plant breeders like Naudin were doing something remarkable. They practiced genealogical selection, they isolated lineages, they measured traits quantitatively, they counted frequencies across generations. They even recognized that different characteristics could recombine independently. In practice, these scientists were treating heredity as something that could be analyzed, tested, and stabilized. And yet, at this point, heredity remained conceptually unstable. There was still no general law, no predictive framework, so that they could not predict what the offspring would be based on the parents, and no clear model explaining why traits would reappear, disappear, or persist. The desired constancy of a trait still relied on repeated human intervention, so on selection carried out generation after generation, rather than on an understood structure of inheritance. So the Vilmorin occupy a great place in the history of genetics because they transformed plant breeding into an experimental science. They pushed heredity towards quantification. They brought heredity to the edge of formulating a theory, but they were still standing just before the threshold. So we've come to the end of this video. In the next video, that will be the final part of this chapter, we will shift perspective once more. Away from animal and plant breeders, we will move into the medical world to see how physicians and medical thinkers grappled with heredity in the human body. Because before Mendel formalized his law of inheritance, heredity was already a pressing question in fields that go far beyond agriculture. So I'll tell you, thank you for watching. If you like following this journey into the origins of genetics, make sure that you subscribe so that you won't miss the final part of this chapter and any other theme that I will deal with in the future. Once again, thanks for watching, and I'll see you very soon. Goodbye.